Okay, so we are recording and welcome everybody. Good morning here in Brazil. Good afternoon in Sweden. It's a pleasure to welcome Dr. Hanna Farnelid. Is that correct, the pronunciation?
Yes.
And thank you very much for accepting to give a talk at our seminar series. And Hanna is going to talk about Picophytoplankton in a changing world. She's a professor at Linnaeus University in Sweden. It's a pleasure to have you here. And hopefully we're going to have some engaging discussions after your presentation. So please, Hanna, start.
Thank you, Eduardo, for the introduction, and thank you very much for the invitation. I'm very excited to meet you here today. And we have history going back several years now with a collaboration with Professor Renata. And two of her PhD students have actually come to work in my group here at the Linnaeus University in Kalmar. So this has been a really great experience with exchange of knowledge and really inspiring both for my group and I hope also for the students and your research environment. So really happy to have this collaboration. And today I will talk about my favorite group of phytoplankton, and those are the Picophytoplankton, the smallest of the phytoplankton. And I will tell you a little bit about the research that we have done here over the years at Linnaeus University in trying to solve some of these mysteries with the Picophytoplankton and what do they really mean in terms of microbial ecology and to the environment as a whole. But first, I would like to start with just showing a picture of where we are located here at Linnaeus University. So this is in southern Sweden, and the city of Kalmar is in southeast Sweden. And it's really an old town with a lot of heritage. So we have a castle from the medieval times. And if you look in just the other direction, we have this super modern university campus just at the waterfront. So this is overlooking the guest harbor. And I'm right now on the fourth floor of this gray building that you see to your left. So I'm also just overlooking the ocean here in the Baltic. And just to make some advertisements, we do have a bachelor program in biology, a master's program in aquatic ecology, and also a PhD program in ecology. And I am leading a research center in ecology and evolution with around 65 co-workers, where we are 11 research leaders. And we are working a lot in the microbial food web, looking at interactions between different organisms, going all the way from viruses to fish and bird species, and looking at how they are interacting in the marine environment. And much of our expertise is here in the Baltic, but I also do quite a lot of research in polar environments, and I will tell you a little bit more about that later. And a lot of the time, the acknowledgement slide comes at the end, but I really want to show this at the start of my presentation, giving recognition to many of the researchers in my research group that have contributed to these studies over the last decade in exploring the role of Picophytoplankton. And we've also been very lucky to be supported by various different funding agencies to be able to do this research. And that has been really fortunate. So let's start by moving out into outer space and looking down at the Earth from space using satellite images. And these images are showing the chlorophyll concentration in the global ocean. And the green, yellow, red colors are indicating areas where there is more chlorophyll. So you can see around the coastal areas in upwelling regions, these are actually hotspots of marine primary production. But large areas of the global ocean are these blue areas, which are actually like an ocean desert where there's not really a lot of nutrients available for primary production and phytoplankton growth. And these areas are of particular importance when it comes to the strategies of picophytoplankton in scavenging nutrients in nutrient-poor environments. And when we look at phytoplankton in the ocean, we have categorized them into different size classes, from the megaplankton to the macroplankton, microplankton, nanoplankton, and the picoplankton. And the traditional net plankton, the larger phytoplankton, are those that we typically sample with a phytoplankton net or and look at using light microscopy. For many, many years, we were more or less disregarding the smallest size fractions because those cells were so small, so we were not really detecting them using these traditional techniques. But around 30, 40 years ago, with flow cytometry applications, these smaller phytoplankton groups were really discovered and found to be very, very abundant in the global ocean. So putting this into context, I want to show you this illustration of different sizes of phytoplankton and how they may contribute to the microbial food web and carbon. So in the microbial loop, smaller phytoplankton and remineralization of nutrients from bacteria is the main mechanisms. And the more efficient trophic transfers are from the larger phytoplankton groups up into the higher trophic levels. And larger phytoplankton, they also have a more tendency to sink faster. So they will also give a stronger contribution to the biological pump and carbon sequestration. But how these smaller phytoplankton are contributing to carbon export or transferring to the food web is not really known. It's typically been associated by more steps in the food web and also more carbon losses. And the traditional view is that these small cells don't really sink out of the water column. In order to sink, they need other mechanisms such as aggregation or having been fed on by a larger organism. So what are these picophytoplankton? Well, it's a group of phytoplankton that are defined as smaller than 2 micrometers in cell diameter. And they perform photosynthesis. The main groups are picocyanobacteria and picoeukaryotes. And picocyanobacteria are primarily made of Prochlorococcus and Synechococcus. And if you look at them under an epifluorescence microscope, it might look like something that you see here on the right, where you see the larger red cells, those are the picoeukaryotes, and the smaller orange colored cells are from Synechococcus. So this is an image of Baltic Sea picophytoplankton. And if we look at these groups of picocyanobacteria and picoeukaryote, they really have a global distribution. And the most abundant photosynthetic organism on Earth, that is Prochlorococcus. And Prochlorococcus in the tropical and subtropical oceans can reach more than 100,000 cells per milliliter. So there's very many Prochlorococcus in our oceans. And they are successful in these waters because they are very good at acquiring that low nutrients that is available in these large areas of the global ocean that are nutrient limited. Synechococcus is kind of a larger cousin to Prochlorococcus, so it's genetically related. And Synechococcus has a larger distribution also into colder and more nutrient-rich waters. But as you can see on this image, it does not quite reach as high abundances as Prochlorococcus. When it comes to the picoeukaryotes, they also have a global distribution, and they tend to peak at the chlorophyll max level, and they are also more abundant in, for example, upwelling zones. And these different groups of picophytoplankton, they are distributed based on different light and temperature regimes. So in the image to the right, you can see the temperature and biomass distribution. And the black denotes the picoeukaryote phytoplankton, while the blue is Synechococcus and the red is Prochlorococcus. And here you can see that the picoeukaryote biomass is higher at the lower temperature ranges, while Synechococcus has a preference range of 5 to 20 degrees, and then also reaching higher abundances up to 30 degrees, while Prochlorococcus is limited to these more tropical and subtropical areas. And one of the reasons why I'm so interested in this group of phytoplankton is that future model predictions are predicting that there will be an increase in the total picophytoplankton biomass. So in a future scenario where there will be more stratification, the characteristics of picophytoplankton will make them more successful. And this is especially true in the northern hemisphere, where we expect an increase of between 10 and 20 milligrams per meter per cubic meter. So what will happen then if there is a shift from larger phytoplankton groups to these smaller phytoplankton? Well, I told you previously about carbon transfer in the microbial food web. So with the larger group of phytoplankton, there's more efficiency in the transfer of carbon into higher trophic levels. There will also be more carbon sequestration because these larger phytoplankton cells sink more efficiently. If instead we have a population of smaller phytoplankton, the transfer in the marine food web will not be as efficient, as there is carbon losses in every step of the trophic levels. And there's also an expectation that there will be less carbon sequestration. So what do we know about picophytoplankton? Well, I told you that they are very tiny, but they are also very widespread. They are extremely diverse, and they are important for aquatic ecosystem, contributing 25% of the primary production. But yet, because they were only recently discovered and haven't been much explored, they are quite understudied in some areas. So in the Baltic Sea, for example, picophytoplankton have not been included in monitoring programs except for the northern parts of the Baltic Sea. And here we find an extremely high diversity of picophytoplankton with more than 2,000 unique ASV sequences in the Synechococcus clusters. And we also see that there's shifts in ecotypes with different gradients across the Baltic Sea. So the Baltic Sea is really a large brackish ecosystem going from low saline water of 3-5 PSU, crossing intermediate PSUs, up to 33 in the marine habitats. So we have gradients in salinity, nutrients, and temperature, which really creates these different niche specifications of different ecotypes. So over the year, I have started to collect different picophytoplankton strains, and here is a picture of just a few of them. In total, I have around 35 different strains from the Baltic Sea. And as you can see, these are really, really beautiful in that they have different pigmentations, and these different pigmentations cause them to be optimized for different light conditions in the aquatic environments. So these green strains, which are containing more phycocyanin pigments, they are more optimal for the red light spectrum, which we typically find in turbid waters closer to the coast, while the red strains are more adapted to the blue light regimes, which we'll find more abundant in the open waters. So we see these really large variation in the pigment composition. And what is interesting is that many of these clades are actually coexisting in the environment. So when conditions are changing, the abundance patterns change according to those environmental conditions. And we have been doing studies on this, both using laboratory experiments and also going back into the environment and trying to explain those environmental patterns by what we see in the lab. So here is just an example of 5 different strains that belong to different phylogenetic clusters that we have done extensive characterization on, looking at how they respond to changes in salinity, temperature, and light conditions. And there's lots of details in this image, but the story I want to tell you is that none of these isolates are responding the same to these different conditions. So some are very, very tolerant to a wide range of salinities, while some are more sensitive to higher or lower salinities. There's also a different range of the temperature optima, where some seem to really have an effect with temperature, and some are not as sensitive. And similarly with the response to different light conditions. And I think this is really interesting because that explains why these strains are coexisting, but it can also tell us something about which strains will be more successful in a future scenario with different conditions in our environment. And I want to also briefly tell you about an experiment that Eduardo did during his time here at the Linnaeus University. And this was a heat wave experiment that was conducted. And heat waves are predicted to become more and more prevalent in this area of the world. And what he did was a quite simple experiment with 3 of these different isolate strains, and then exposing them to a heat wave condition for 7 days. So the heat wave exposed cultures were exposed to 27 degrees, and then there were also control cultures that were kept at 18 degrees. And what is really unique in this study was that he also did a recovery phase to bring all of the cultures back to 18 degrees and see if there was a difference in the recovery after this heat wave exposure. And Eduardo can tell you a lot more details about this results, but I just wanted to show you the big picture in that none of these strains are responding the same. We have 3 different strains, and one does not really seem to have an effect of a heat wave at all, while the other one is affected by the heat wave, but not in the recovery phase. And the third isolate is significantly impacted by the heat wave treatment and also into the recovery phase. So for some strains, there seems to be quite an extensive adaptation to this type of heat wave, while for other strains, they are significantly negatively impacted. So this will affect which strains will be successful in a future climate when we will be more exposed to heat waves. So over the years, so over the past 10 years or so, my research group and I have been doing quite extensive samples, both in the open water and in more coastal areas here close to the Linnaeus University. And we have been looking at abundance patterns, diversity, and also doing isolations for this Kalmar algae collection. And during these years, we've been able to study the patterns and the abiotic factors that are regulating the abundance patterns of Picocyanobacteria in the Baltic Sea. And in this image, you see the red dots are showing the abundances of the phycoerythrin-rich strains, so the red pigmented isolates, and the blue ones are showing the green strains. So this is environmental data, but you see this really clear reoccurring pattern with peaks in the summer season of abundances of 10 to the 5, 10 to the 6 cells per milliliter. And we also find that at this coastal station, we find more of the green pigmented strains. And that is very much in agreement with the theory about the environments where these strains are selected for. And we see this clear correlation pattern both with temperature and nutrients. So with higher temperatures, we also see increased abundances of the Picocyanobacteria. So the photosynthetic picoeukaryotes is a group that has not been traditionally studied very much. But these studies are really showing us that they are a significant part of the biomass. So I want to draw your attention to the bottom panels in this graph. And this is showing the contribution to the total phytoplankton biomass. And the green parts in this picture is the picoeukaryote population, and the purple is then the other phytoplankton. And when we think of phytoplankton traditionally, we think about the diatoms, we think about the filamentous cyanobacteria that are forming really extensive bloom. But when we add these smaller phytoplankton groups, the picture that we see is really quite different. In that although these very, very small cells seem negligible, when we add them all together, the total biomass that they contribute to is not at all negligible. These are really composing a large part of the phytoplankton community, frequently more than 50% of the total biomass. So where this biomass ends up, whether it is transferred up into higher trophic levels, whether it is sequestered into the deep ocean, it matters. And today, we don't really know these different pathways and how important it is for the different processes. So this is something that I'm really, really eager to find out more about, where this huge biomass actually ends up. So we've done quite a lot of work with the picophytoplankton and the abiotic controls. We find that it's positive correlation with temperature. We see these recurrent patterns. We see shifts in community composition. We have negative correlation with nutrients. And what I'm eager to know now is more about how the biotic controls are really affecting these picophytoplankton community. So that's the direction that the research here in the Baltic Sea is heading. But now for the next section of my talk, I would like to zoom out a little bit and talk about global warming and changes in the Arctic Ocean. So we have all seen this image of rising temperatures on Earth, with a steady increase of temperature across the globe in comparison to pre-industrial times. And on the right-hand side, you see the increase in temperature plotted at different latitudes. And if you look in the bottom right-hand corner, you see this red or reddish-brown color. And that is the area on Earth that is warming the fastest. And that is the area around the Arctic Ocean. So the Arctic Ocean is warming 4 times faster than the average on planet Earth. And the increase in air temperature has already reached beyond 3 degrees Celsius. So this is a process called Arctic amplification, where the Arctic is warming faster than other areas on Earth. And there are multiple explanations to this. One is the ice albedo feedback loop. And that is something we all can relate to. Like if we go outside on a hot summer day wearing a white t-shirt compared to a black t-shirt, we will feel the temperature difference by the absorption of white color compared to a black color. So as the sea ice is melting and there will be more open waters, those open waters will be absorbing more of the heat compared to the sea ice, which was reflecting more of the heat. So this means that the more ice that melts, the more heat will also be trapped in the Arctic. In the Arctic, there is also heat transport from lower latitudes. So if this water is warmer, then that warmer water will be increasingly transported into the Arctic, which brings more heat into the Arctic. So that also accelerates the heating. And because of the characteristics in the Arctic, there's also a lack of strong convection, meaning that the heat that is in the water is actually trapped near the surface. And those are several reasons why this Arctic amplification occurs. And an effect of this Arctic amplification is that there's a very rapid sea ice decline. So in this image to the left, you can see the sea ice decline from 1980 to the mid 2020s. And the sea ice is actually more than halved in the sea ice area. And that is really clear also from satellite images that you see here to the right. And it's not only the sea ice extension, it is also the volume and thickness, and multi-year sea ice is becoming more and more rare. So this means that the ecosystem that is the largest of the Arctic large marine ecosystems, the Central Arctic Ocean, which has been characterized by both permanent ice cover in summer and winter, and is about 7 times the area of Sweden. That means that this ecosystem is rapidly changing. So with the increasing ice melt, there will be changes in this permanent ice cover to more seasonal ice cover. With the ice melt, there's also going to be more freshwater habitats or brackish water habitats with the meltwater mixing into the marine water. And this is also causing more stratification and changing the availability of nutrients. And this will then affect the biological production. When there's less accessible nutrients, primary production will eventually go down. And this will in return also affect the distribution and composition of species. So in this image from the National Geographic, you really see the phytoplankton at the bottom of the food web and the zooplankton supporting these animals that we typically associate with the Arctic Ocean. But again, as always, it's not the phytoplankton and the zooplankton that most of us are giving our attention to. But in this changing ecosystem, the base of the food web and how that will be affected in a future climate is really a key to understanding how these ecosystems will look into the future. So we can expect changes in the phytoplankton composition. So if we look to the left in this illustration, we have ice coverage where we have diatoms forming under ice blooms. And there's nutrients from the ice. There's quite limitation in light, but this is a really good habitat for the ice-associated larger phytoplankton. In the marginal ice zone, when the ice starts to melt, that will also increase the light penetration, causing more larger phytoplankton to form blooms. And this is typically what will occur in Arctic spring blooms when there's high productivity. If we move more into the open water, the water column is more stratified. That means that there's less mixing, so less nutrients in the upper water column. And that causes smaller phytoplankton to be more benefited. So the phytoplankton community will be more dominated by smaller phytoplankton. And when we went into the Central Arctic Ocean and looked at the chlorophyll concentration, we found consistently low chlorophyll concentration. So if we compare this to the Baltic Sea, here we typically see this greenish bluish color, and that is because we typically have quite high production. There's lots of nutrients from agriculture, so we tend to have higher production. In the Central Arctic Ocean, the color of the water is really, really blue, so we have low productivity, and we really saw a low production in the upper 50 meters or so. And we did size fractionation to look at how much chlorophyll was in the large size fraction compared to the smaller size fraction. And we found here that, so you see in the graph with the yellow bar, that's the microplankton fraction. And the pink bar is showing the picophytoplankton fraction, where the smaller phytoplankton were really contributing 75–80% to the chlorophyll biomass. So this is really showing us that picophytoplankton are the major contributors to phytoplankton biomass in the Central Arctic Ocean. And if we look again at this image in the bottom, you see the contribution of the small size fraction to the total chlorophyll, and these red colors are showing more than 80% contribution. So over this 2,500 kilometer cruise track, there was only one measurement where larger phytoplankton groups were dominating the phytoplankton community. And similar to what I have done in the Baltic Sea, during 5 different expeditions to the Arctic Ocean, I have started to build a Arctic culture collection, and we now have around 25 strains of Arctic cultures that we are keeping and studying here in the lab. And we are looking at different ecotypes and looking at how these strains are affected by different environmental conditions. And I want to show you one experiment that a bachelor student has recently performed, and that is growing a strain of a pico-eukaryotic algae called Micromonas polaris, which is the dominant pico-eukaryote in Arctic waters, and it typically consists of more than 50% of the biomass in many of the samples we have seen. And we have also found that it's really abundant in fjord systems in northeastern Greenland. So what the student Moa did was that she took these cultures and she grew them in medium of different salinities. So the strain was originally isolated from around 33 PSU. And she grew it both at higher salinity at 35, but also at 30, 25, 20. And what she found was that the fastest growth rates that she observed was actually at 20 and 25 PSU. And this suggests then that this really abundant strain could be even more successful in a future Arctic Ocean when there's more influence of meltwater from glaciers and from sea ice, that this will also be beneficial for this pico-eukaryotic strain. And moving into the Arctic, one of the hypotheses that I've had on these expeditions is that there might be Synechococcus entering the future Central Arctic Ocean from this Atlantic water inflow. So this is a process called Atlantification, and Synechococcus has been suggested to be an invasive species of the Arctic. And there was a study published in 2016 which actually showed Synechococcus north of Svalbard. So on my expeditions to the Arctic, I've been looking for Synechococcus to see if there's an indication of this Atlantification. But so far, I have not been able to detect Synechococcus. So this is an example of a transit from Sweden up to north of Svalbard, both going north. In the orange dots are showing the transit to the north, and then the green dots are showing the transit south. But on neither of these transits, we have been able to find Synechococcus north of 76 degrees. And I find this really interesting because I don't doubt that the previous study had found them, but we don't find any evidence of them establishing in the Arctic. But this is an example in science where these negative results are perhaps not as impactful as if it had been positive results. Because should we have found that these Synechococcus were really establishing themselves in the Arctic, that would have been a scientific paper that would be more impactful. And for now, I just have these results in my drawer, but I also want to share it with you as an example of these negative results that might not ever get published. So, Picophytoplankton in a Changing World is the title of my talk today. And we are finding that the abundances and the composition of these different ecotypes in different habitats is this complex interplay between both the abiotic and biotic factors. And I really want to stress these interaction effects and selectivity when it comes to grazing or viral lysis as something that we really need to find out into answering these questions and how these abundance patterns are truly regulated in nature. And if you did not know it before, Prochlorococcus, the most abundant photosynthetic organism on Earth, it's really here to stay. Picophytoplankton are really adaptable, and they are also expected to be very successful in a warmer ocean. And I really think that in this context, we need more knowledge in order to understand how these shifts towards smaller phytoplankton will really affect ecosystem productivity, carbon sequestration, and services to us humans. So to end this talk today, I also want to talk a little bit about my current plans and what I am working a lot on right now. And that is a little bit different from this core research in that I am coordinating an early career research program on the icebreaker Odin. So these are the happy students that I was teaching last year. And this is a PhD course on the Arctic Ocean climate system. And last year we had 18 PhD students from various different countries. And in this course, we are combining practical field work and giving lectures and also doing workshops on board that the students are discussing issues related to Arctic research in the future. And we also have a blog on the Swedish Polar Research Secretariat webpage. So this year, I will be returning to the North Pole for the fourth time. leading this research school in the Arctic. And this year we have 25 students enrolled, and we will go for a 6-week expedition to the Central Arctic Ocean. And I really think that this is a unique experience for the students to really get wet, to get their fingers on the science, where it happens. And I think that is so important because we are working now more and more with just computational analysis models. But if the students can get out into the environment, do the measurements, see where the data comes from, then they can understand the ecosystem more as a whole. And another key is these collaborative efforts, which is really, really important in research today. And coming together from different research fields, we can really both get synergistic effects to do excellent science. But that's also where the creative ideas happen when we are talking to someone that has a different perspective. And that's what we are doing now as well. I'm telling you a little bit about the Baltic Sea, and then I can learn about the things that you are thinking about in your fields of research. And I also want to encourage both creativity in science, but also expression, that we speak up and perhaps not only speak science. Many of us are experts also in music or dancing or other art forms, which we can combine with science to make more people understand and make more people aware of the things that are happening to our planet Earth. And this is one of my favorite pictures. This is from the North Pole and my team from the Linnaeus University back in 2021. And I want to take just 1 minute to tell you about the North Pole because it's quite a special place. So standing on the North Pole and standing on sea ice with 4,000 meters of seawater underneath of you is quite a different experience. And thinking about the planet and thinking about what we are doing to our planet. But it's also a really fun place. You can choose any time zone. So I could be in Brazil right now in the time zone because you can just turn to whatever time zone you want. And there's only southern winds at the North Pole. They may be cold, but they are always southern. So it's quite a special place. And my last slide here is an image of the first sunset when leaving the Arctic. And you can see this polar bear wandering, and there's this path of melt ponds that the polar bear is crossing. So this is the melting sea ice forming little lakes on the ice as the polar bear is walking away into the sunset, and we are leaving the Arctic. So with that, I would like to end the presentation part of the seminar. And I understand that there will also be some opportunities for discussions and questions. So I'm very excited about that.
Well, thank you very much. This ending was quite something. Excellent seminar. Let me just stop the recording, but thank you so much. Congratulations for the talk.